Good morning, students. Next, we are going to discuss in biobotany 12th plant tissue culture, type of the plant tissue culture. Previous class we studied tissue culture, technique involved in tissue culture. You know this different type of the technique will be there. Technique means sterilization, media preparation, culture condition, callus formation or callus induction, organogenesis and hardening. This is a technique involving tissue culture. Next is a type of the tissue culture. Here this type of the tissue culture based on the explant is a very important. Our type of the type of this plant tissue culture is based on the explant. We can classify it into four type of this plant tissue culture will be there. One is the organ culture. Second one is a meristem culture. Third one is a protoplast culture. Fourth one is a cell culture. About these four type of the tissue culture will be there based on the explant. What the explant we are using. For example, organ we are using the plant organ. That is the organ culture. We are culture the apical shoot, meristem, root meristem. That is we call for the meristem culture. Or these protoplast we can isolate and cultivated the protoplasm culture. Or the cell culture we isolate the cell from the tissue. We have cultivated the cells. We have cell culture. This we have to say this one to one. First is the organ culture. But this organ means first what are these organ? This organ means the embryo, root, shoot, anther, ovaries. That is organ of the plant we are using for culture. For example, organ culture, the embryo, root, shoot, anther and ovaries. You know this is the organ of the plant. We can use for the culture this media or use culture media or culture technique. We can produce the new plant. That is the organ culture. You know this is visually, the diagrammatically we can understand the organ culture. The organ culture we can first explant, take the leaf we can get the cell tissues from the leaf, sterilization. After this inoculate the culture medium and this formation of this callus formation, rising callus, rising production, rising of this plant will be plantlet we can produce from that plant. Next this plant will be gardening. But this organ is you can use for the culture of this plant, organ culture. What are the organ we can culture? Two more questions. Ask two more questions. What are the organ involved in the tissue culture? Embryo, root, shoot, anther and ovaries. Another one is a meristem culture. The meristem culture, this tip part of this tip of these plants. You know this two tip will be there. One is the upper part and lower part. And lower part is this root tip will be there. The shoot part will be there. This is shoot tip. After that we can take this explant based on the meristem. This meristem we can take the shoot part of this plant and remove all the matured leaves, and these matured leaves remove that apical tip part we can transfer to the inoculation, the culture medium. There is a callus formation. Next the callus to the shoot, root formation, new plantlet. This is a very important thing. The meristem culture, why we want the culture meristem means the meristem culture used for the protection of the resistant plants. Virus resistant plants we can produce from the meristem culture. About meristem, one of these type of this culture. The organ culture. Organ culture means organ is used for the culture of the plant. This produce the whole plant from the organ. Next, this meristem also is used for the plant shoot formation. This is organ culture and meristem culture. Next, protoplast culture is a very important third culture, protoplast culture. So the protoplast culture. But third protoplast means, first we want to know that protoplast. Protoplast means the protoplast is one of the cell. The cell, protoplast is a cell. This without the cell wall. Protoplast is a cell. The cell without the cell wall bounded by the plasma membrane. You know this plant cell. Have these cell organelles, nucleus will be there. But this remove the cell without the cell wall. Only have this plasma membrane or the cell membrane. But this protoplast means protoplast is a cell without the cell wall. Only found in the cell membrane or plasma membrane. That is we call for protoplast. But this protoplast, the term, the protoplast, the term was introduced by Einstein. Who was introduced the term of the protoplast? Einstein, 1880. One more question. Einstein introduced the protoplast. It's not given our book. It's extra information. Next, this protoplast, using isolated protoplast or using the protoplast, used to produce the whole plant. Produce the whole plant. Have these totipotency. This protoplast have the totipotency in the plant tissue culture. In this biotechnology, the plant protoplast have this totipotency. Have the ability to produce the entire plant from the single cell. The single cell produce the whole plant. But define protoplast. Protoplast is a cell without the cell wall. It is bounded by the cell membrane or the plasma membrane. It is used as a ability to produce the whole plants from the single cell. And also protoplast is used as a produce a somatic or develop the somatic hybrid. Is a very important tumor causing. What is mean by somatic hybrid? You know this body hybridization or somatic hybridization. We can produce the hybrid from the two different parents or two different cells. But here the protoplast is able to differ from this other culture. Here we can produce the hybrid plants. There is the tissue culture is a varied from protoplast culture. Tissue culture, what plant we can give? We can transfer to the media. The plant will be developed. The same plants, number of the million of the plants we can produce. But the protoplast culture, we can produce the hybrid plants. This is a very important thing. Hybrid plants. Here we are using the somatic cell. We can produce a somatic hybrid from the different cells to different cells. This is a very important tumor cuisine. Define what is protoplast. Next, we can isolate the protoplast or this protoplasm culture. We have this number of the steps will be there. First, we have this 4 step for isolation of this culture of the protoplast. One is the isolation of the protoplast. Second one is the fusion of the protoplast. Third one is the culture of the protoplast. Fourth one is the selection of the somatic hybrid cells. Selection of the somatic hybrid cells. It's a very important steps will be there. Important 5 mark question. Sometimes is a 2 mark question will be there, and this 3 mark question also will be there. In this topic, in this part is a very important in plant tissue culture. Okay, these steps will be there. These 4 steps will be there. One is the isolation of the protoplast and fusion of this protoplast, culture of this protoplast. Another one is a selection of this protoplast. The first one is the isolation of this protoplast is a very important. Here, isolation of this protoplast will be the two step. Two methods will be there. One is a mechanical method, another one is a enzymatic method. In our textbook, only for the enzymatic methods only given. This mechanical method. First, which method is isolate the protoplasm? Mechanical method. Who? Clerk 1892 first isolate the protoplasm from mechanical methods. Mechanical. Here, sometime ask you a question. What is how can he produce the naked protoplasm? Two methods will be there. One is a mechanical method, another one is enzymatic method. First method, enzymatic mechanical method is here get the poor yielding of the protoplasm. This is not followed for. Next, this enzymatic method. This enzymatic method introduced by Hawking 1960. This is important thing. Enzymatic method. Isolation of this protoplast based on the enzymatic method. Mechanical method we are using only for this knife physical method. This enzymatic chemical method isolation of protoplast or enzymatic method of the isolation of the protoplast. Sometimes ask different question from that part. This enzymatic method is introduced by Hawking 1960. First, this enzymatic method. I wrote this flow chart. Easily we can write for exam point of view. How can the steps the enzymatic. First we want to take this 10 week old leaves. If we can select first protoplast isolation enzymatic method. First we want to take the 10 week old leaves. Matured leaves, small bits of the leaves or plant tissues or part of the plant. Next, this leaf tissue immerged or stabilization. Before of this immerged of this picker containing, we can stabilize the leaf. Stabilization is a very important thing. The leaves we can take and stabilize. We can select the leaf and stabilize the leaf. After that, the leaf tissue immersed in the enzyme container. Very important. Enzyme. What are the enzyme container? The speaker or any conical plates or any petri plates, any glasswares. The speaker containing or these glasswares containing the leaf immersed in the enzyme container. This enzyme, what are the enzyme? Will be there. One is here, very important two more question. What are the enzyme will be there involved? First one is a 0.5% of this macrozyme. The macrozyme are 10. Another one is here 0.2% of the Onasuga cellulase enzyme. Another one is 30% of sorbitol or mannitol. And this pH, this solution this medium pH will be the 5.4. This is important one more question. How many percentage? 0.5% of the macrozyme, 0.2% of the Onasuga cellulase enzyme, or 30% of sorbitol or mannitol. Sir, this is this enzyme. What is function of this enzyme? First, the 0.5 macrozyme. This enzyme. We are obtained from the Rhizopus fungus. This is a very important Rhizopus fungus. This macrozyme is used for the degrade the cell wall. Degrade the cell wall is an important one. Another one is the enzyme is a cellulase. This enzyme is not only for the single. This is also not only single. Macrozyme have the high pectinase and hemicellulase enzyme. Here this cellulase also will have this mixture of this enzyme. This pectinase, hemicellulase and cellulase is used for the degrade the cell wall. This is a degrade. This is cellulose. You know, in the plant cell is made up of cellulose, pectinase, hemicellulose. You know the cell wall. This cell wall we can degrade the using cellulase and macrozyme. this is a isotonic solution. Charpita or Anita, we can this 30%. This medium pH will be 5.4. Is a very important 2 mark question. What's the enzyme containing this enzymatic method? Another this solution. First we want we can visible or see the diagram picture given our textbook. We can see easily, we can understand. Next step, after that emerged, here what will happen? This plant leaflet. What will happen? This plant the leaf epidermis will be peeler. தோல் உரியும். இப்போ உரிஞ்ச பிறகு என்ன இருக்கும்? Plant cells only there. Next, these cells will be this separate. The plasma lysis will be take place away from the protoplast, away from the cell wall. Upon next, this cell wall will be removed using the enzyme container. First, we want to take the leaves, sterilize the leaf. Next, the sterilized leaves we can peel the epidermis will be peel off. Next, we can immerse in this leaves will be take place. Everything will be happen the enzymatic method. After this enzymatic method, we are incubated. We are immersed in the container and this incubated for the overnight. Overnight full of only 25 degrees Celsius. Overnight we are kept it. After we are gently teasing the cell, light we can that. Next we can transfer to the 20% of the sucrose. Very important 1 mark question. Next, this protoplast solution, the protoplast transfer to the 20% of the sucrose solution for retain their viability. Very important 1 mark question. Here the sucrose used for how many percentage? 20%. What? Retain for the viability of the protoplast. Retain the viability. Next stage, this protoplast we can centrifuge. Centrifuge we are getting the pure protoplast and debris we can remove. But the protoplast we can isolate. Protoplast isolated. How can you protoplast isolate? If the 2 step will be there, one mechanical method, enzymatic method. Mechanical method we can get this poor yielding, but the enzymatic we are getting this more using the following steps. This is a very important. Sometimes ask 5 mark question. How can you protoplast isolate by enzyme method? This is a very important 5 mark question. 1 mark question will be the fecal containing solution. Sometimes ask 2 mark question or 3 mark questions. Next, second protoplasm culture isolation. Second fusion of protoplasm. First, isolate the protoplast. We know the mechanical or enzymatic method. Enzymatically, we can isolate the protoplast. After the purified this protoplast, we will use for fusion of the protoplast. About the isolated or purified protoplast, we can take in the Petri plate. There we are using the fusogen agent. And the fusogen agent we call for the Beck. Polyethylene glycol. Very important 1 mark question. Fusogen agent. Which one of these fusogenic agent in protoplast? Polyethylene glycol. 1 mark question. This polyethylene glycol used as a fusogenic agent. This next the isolate the protoplast. We can incubated the 20 to 30% percentage concentration of the polyethylene glycol plus with calcium ion. What happened? This will be the agglutination of protoplast and fusion of the protoplast. Is a very important. What happened? Agglutinase திரள்தல். ஒன்று எல்லா தனித்தனியாக இருக்கிற ப்ரோட்டோபிளாஸ்ட்னா என்னது ஒன்றா வருது, எல்லாம் வந்து சேரும். அப்போ அக்ளூட்டினேஷன், அதான் ஃபியூசன் சொல்கிறாங்க, இல்லை ஃபியூஸ் ஆகுது. இந்த அக்ளூட்டினேஷன் அதர்வைஸ் என்ன சொல்கிறாங்கன்னா the formation of the clump of the cells ன்னு சொல்கிறாங்க. The clump of the cells னா is a micro, otherwise we call for the micro colony. அப்போ the group தனித்தனியாக இருக்கிறது வந்து என்ன ஆகுது, தனித்தனி ப்ரோட்டோபிளாஸ்ட் will be combined ஆகுது. combine form, that is we call for the clump of the cells in way clump தனித்தனியா இருக்கிறது ஒண்ணா வந்து சேரக்கூடிய அந்த process தான் நம்ம agglutination and fusion what will happen. அப்ப fusogenic agent we are using the 20 to 30 degrees Celsius, actually 25 to 30 degree concentration of the Beck and the calcium ion. Agglutination is a very important. Next, after third step, after this fusion of this protoplast, we can use the culture of this protoplast. Third step. Up here, this MS medium we are using for the culture of the protoplast. Here, MS medium some modification will be there. Here, MS medium with the droplet plating or micro drop array is a very important. It is a droplet plating. You know this Petri plate. Here, take for this medium. Here, not take for the medium. First, we can put in the protoplast. After that, this medium will be dropping. What is essential? What is essential? We are putting the droplet medium. There is a modification. The protoplast, this culture, tissue culture, we can medium. First we are taken medium. After the transfer the explant. Here, take the protoplast after we are dropping the medium. That is a modification with the droplet or plating micro drop array means we are take a single protoplast culture. Single we can take for within this Petri plate we can grow for the separate. We have the separate Petri plate or dishes will be there. We can cultivate the protoplast. That is we can modification droplet plating or micro drop array. Different method. Next, before of that we can using for the viability test for protoplast. This protoplast, isolated protoplast have this viability. It's not it has the viability or not. We can find out using for fluorosin diacetate. This is used for the viability of the viability test of this protoplast. Very important one more question. Before of that, isolation of the protoplast, retain the protoplast, retain the viability. We are using the 20% of sucrose. But the test of this viability, we are using the fluorosin diacetate. This is important one more question. After that, incubate the protoplast 1000 to 2000 lakhs light with 25 degrees Celsius. We can incubate. After that, the cell formation will be happening. And first, this protoplast without the cell wall. First, the culture in this cell wall formation will be happening. But before of this culture, we are test is a very important. Viability test is a very important part. We are using the fluorosin diacetate solution. Next, incubate 1000 to 20,000. 1000 to 2000 lakhs light is essential for 25 degrees Celsius. After that, the cell wall formation 24 to 48 hours. How many hours cell wall formation will be take place? 20 to 48 hours is need for the cell wall formation. Next, new cell. When this new cell formation, after this 2 to 7 days, the new cell or the cell division will be take place. Our single protoplast will be divided into two cells when will be take the 2 to 7 days after the cell culture after the fusion of protoplast, the culture of protoplast. Third, fourth one is here culture of this protoplast we can MS mediate transfer control. MS mediate with modify with the droplet plating or micro droplet array method. Before of this culture we are test for viability. We are incubate the protoplast for 1000 to 2000 lakhs light and 25 degrees Celsius. Next, the cell wall formation is a take for 24 to 48 hours. And the new cell or cell division will be take place 2 to 7 days. Next, fourth one is a selection of the somatic hybrid. This is a very important. Last public look at the two opposite hybrid. This is a very important difference between hybrid or hybrid. Do you know this hybrid? Hybrid means fusion or produce the hybrid from two different cells or two different parent. Produced from two different parent. That is we call for hybrid. hybrid also we two different, but the hybrid what is the main difference? This hybrid means after the protoplast fusion, only the cytoplasm fusion without the nucleus from the different cell. Different protoplasm will be fused without the nucleus. That is we call for hybrid. Without the nucleus, only fusion of the cytoplasm from different cell. Cytoplasm of different cell. This is a very, very important. Cytoplasm or protoplasm of the different cell. Only fusion of two different protoplasm. That is we call for hybrid. Next, after hybrid, the nuclear fusion will be take place. That is we call for somatic hybrid. What is the difference between hybrid and hybrid? Hybrid means two different cell fused only for the protoplasm without the nucleus. With the nucleus we call for hybrid. Without the nucleus we call for hybrid. This is very, very important. Last public 2 mark question asked the hybrid. Define hybrid. This is a very important 2 mark question. What is hybrid? What is hybrid? This is an important 5 mark question for protoplasm culture. Protoplasm culture. Next, fourth plant type of the tissue culture, cell culture. Cell culture or cell suspensor culture. What's a cell suspensor culture? Cell suspensor means is a hanging of the cell. What is the difference from the protoplasm culture and the cell suspensor culture? There is here medium is a solid nature. Here is a medium is a liquid in nature. We can the cell we can suspended the medium or this hanging in the medium. The cell suspensor culture. What is meant by cell suspensor culture? The cell is growing of the cells in the culture medium. Here single cell or aggregated cell in the in vitro culture medium is known as the cell suspensor culture or the cell culture. Is a 2 mark question. What is meant by cell suspensor culture means the growing of the cells including the culture of the single cell or aggregated cell in the in vitro culture. In vitro culture in the liquid medium we call for this the cell suspensor culture. How can you prove for the cell suspensor culture? We can prove by this the callus. The callus we can take this medium. We are kept in this medium. Here, this callus cell suspensor is performed by transferring a portion of this callus in the liquid medium. Transfer the callus of this liquid medium. There we can agitated. What is mean? Agitated. We are using the Sacker instrument. Like this more method. இப்போ நம்ம எப்படி தயிரில் வந்து மொத்த போட்டு மோர் கடைஞ்சாலும் சடசடன்னு சுற்றும். The same way, this callus portion of the callus will be agitated by using of the Sacker instrument. But here, this callus will be separated the cell. This single cell separation of. The callus and the cells will be separated from the callus. That is we call for the suspensor culture. We are using the Sacker instrument for the separation of the cell in the liquid medium. That is the cell suspensor culture. This technique is used for secondary metabolic product. Production of the secondary metabolic product in the cell suspensor culture will be used. The secondary metabolic means already you know the secondary metabolic product. Primary, the plant, there is a chemical substance used for the growth and development for living organisms. That is a primary. The secondary means is not used for normal growth and development. That is we call secondary metabolism. These secondary metabolism like alkaloids, flavonoids, terpenoids, phenolic compound and recombinant protein. We have this secondary metabolism. We are product from the cell suspensor culture. Here the secondary metabolism is a chemical substance the substance we are obtained from this living things. This is not used for the normal growth and development of these plants. What is the use? This is used for this byproduct of the cell metabolism. Is used for the byproduct of the cell metabolism. Is an example. One of the example given our textbook is an example for the biosynthesis and isolation of the indole alkalides. Indole alkalides from this Cathanthus rosea. You know this Vinga rosea, Sudugattu malli. That plants used for the indole have this indole alkalide. The alkalide used for the anti-cancer property. Anti-cancer properties obtained from this Cathanthus rosea. You know this biosynthesis. The synthesis produced from the living organism. Biosynthesis. Indole alkalide from the Cathanthus rosea, common name Vinga rosea, and in Tamil we call the Sudugattu malli. One more question. About this biosynthesis. About one of these byproducts of these suspension culture indole alkalides. Indole alkalides. Next, the process of this product from the secondary metabolism scaled up. Scaled up mean அதோட நிலையை கொஞ்சம் உயர்த்துறது. எப்படி? How can this improve the secondary metabolism? Where we can commercial purpose, we are in a bioreactor, using the bioreactor. You know this one of the vessels, large size sterilized vessel, we can produce the large amount of the commercial product. But this we are using for the bioreactor. This bioreactor is we can produce the large amount of the secondary metabolism. Of this secondary metabolism we can produce, many strategy will be there. Many strategies such as one is biotransformation. It's a very, very important. Another one is elicitation. Another one is immobilization. Immobilization. The three strategy is a very, very important. Many strategy, one is biotransformation, elicitation and immobilization of the secondary metabolism. What is biotransformation means? We can transfer this, the chemical changes will be occurring in the this process. Chemically the process will be changed. This elicitation means we can discover what will there in the medium, or what then this produced from the cell, or this aggregated cell. What is we can bring it from the what it will be there. Next we can this immobilize that chemical compound. என்ன பொருள் இருக்கோ அந்த பொருளை மாத்தணும். இப்போ ஃபார் எக்ஸாம்பிள் ஆல்கலைடுனா இப்போ the plants will be produce a lot of these alkalides. அப்போ this which one of alkalide flavonoid will be there in plants. Any one for alkalide we want this plant alkalide. All this we can produce alkalide. The alkalide will be discovered and the discovery will be a permanent. There is a immobilization. நிலை நிறுத்துதல், வெளிக்கொண்டு வருதல், வெளிப்புணர்தல். அடுத்தது கெமிக்கல் சேஞ்ச், வேதி மாற்றத்தை உருவாக்குறது, அதை வெளிக்கொண்டு வர்றது, அதை நிலைநிறுத்துதல். இந்த மூன்று ப்ராசஸ் தான் நம்ம என்ன சொல்றாங்கன்னா இஸ் a main stage. Sometimes also two mark question. What are the strategy will be there in the suspension culture? One is a biotransformation, elicitation and immobilization. Is a very important for the two mark question. In the this process to make the cell suspension culture for more efficient is a very important. More efficient in the production of the secondary metabolism. More efficient. Which one of these strategy will be more efficient for the suspension culture? Which one of these strategy? Biotransformation, elicitation and the immobilization. It's efficient for this more production of the secondary metabolism. Is very, very important. And there is some of the secondary metabolism products given our textbook. Is a very, very important 1 mark question and 2 mark question. One is here digoxin. The digoxin is a secondary metabolism we obtain from the Digitalis purpureum. The plants, these plants produce this chemical compound is used for the cardiac tonic. இதய தசையை வலுப்படுத்தக்கூடிய, இதயத்தை புத்துணர்ச்சி செய்யக்கூடிய டானிக் obtained from the Digitalis purpureum plants வந்து is used as a chemical compound we call for the digoxin. Another one is codeine, is used for Papaver somniferum, obtained from the Papaver somniferum plants, is used as an analgesic, is a வலி மருந்து, வலி நிவாரணி அப்படின்னு சொல்லக்கூடியதான் analgesic. One more question. Next, capsaicin is a capsicum anum obtained from the plants of the capsicum anum. It is used for the romantic pain treatment. மூட்டு வலி மருந்தாக இது பயன்படுது. romantic pain treatment இது பயன்படுது. அடுத்தது vincristine is a Cathanthus rosea. We are obtained from the Cathanthus rosea, used as an anti-cancer property. புற்றுநோய்க்கு எது மருந்தாக பயன்படுது. அடுத்தது இம்பார்ட்டன்ட் quinone. Already you have studied. Quinone is a Cinchona officinalis. Is used for the anti-malarial drugs. Anti-malarial. This is a very important 1 mark question, 2 mark question. Sometimes also 3 mark question, 1 mark question. This is a digoxin obtained from the name of the plant. The digoxin is used for cardiac tonic. This is a very important 1 mark question, 2 mark question. This is a cell suspension culture. Next we can regeneration of this plant. Okay. Next recall from the just now you can see that type of the plant culture. The 4 type you know that one is the organ culture, second one is a meristem culture, protoplast and a cell culture. Just now we have discussed. Again just we can recall for the organ culture. You know this organ we are used for the culturing. Type of this, what are these organ we can use for the culturing? You know this root, stem, leaf, that is we can use for the culturing of this plant, that is we call for the organ culture. What are these organ? Sometimes 2 mark question as for the organ culture. Right, the diagram is very important. You can see this diagram, and whenever we can attend the or watching the class, we can refer our textbook. Keep it your textbook with you. That is a very it is used for reference for you. Next, meristem culture. Second, organ culture. You can culture used of this plant organ, part of this plant, leaf, stem, leaf, embryo, root. We can use for the culturing. Next, the meristem culture we can use for the meristematic zone of the plant. You know this root meristem, shoot meristem. Maximum this plant culture we are using only for the shoot meristem. Shoot meristem culture, that only is here have this virus resistant. We can produce the virus resistant plants that we are using for the shoot meristem culture. That is a next culture is a protoplast culture, very important. Sometimes this is a 5 mark question, sometimes the 3 mark question, 1 mark questions asked from protoplast culture. Very important 5 mark questions. You know this what is mean by protoplast? What is mean by protoplast? What are this method we can isolate the protoplast? I told that 2 method. One is a mechanical, another is a enzymatic method. You know this mechanical method, it's not given our textbook. Mechanical method we can use for the fine knife. Is a mechanically we can get this protoplast, but this get the poor yielding. We are get the poor protoplast. That method we can not use for nowadays. We are only for the enzymatic method only we are using for the protoplast fusing method. That is a very important. What are the steps we can? Then first we want to this isolate the protoplast, fusion of the protoplast. Next is the protoplast culture, and we can select the somatic hybrid cells. That is a step of the protoplast culture. This is enzymatic method, isolation of protoplast. Important 3 mark question. We can know that the step we want first isolate the plant. We can take the plant and immerse the enzyme containing. We can get that. After that, we can use for the sucrose 20% for the viability testing, viability of the or they maintain the viability of this protoplast. Next is centrifuge, that is the steps is a very important 3 mark question. And this protoplasm fusion the enzyme, fusogenic agent, which one of the use for the fusogenic agent? Important 1 mark question. Usually the Beck is used for the fusogenic agent polyethylene glycol. Is important 1 mark questions. Next culture. Before of this culture, there is a top array. And top array method, the culture medium we can modify. We are using this viability test, the pollen viability, the protoplast viability test. We are using the fluorosin diacetate, the chemical for the testing and maintain the viability sucrose, and there is a testing for this fluorosin acetate for the test of this protoplast. Is important 1 mark question. The temperature, what the temperature, whenever this after this temperature, when the protoplast will be cultured or divided in 2 cells, how many days, how many hours will be take, how many days will be take for the formation of this protoplast culture. Next is a cybrid, is a very important 2 mark question. Last 2 mark question, the cybrid. You know this cybrid without this nuclear fusion, 2 different protoplast fused without the nuclear fusion, that is we call for side. After that this nuclear fusion form, that is we call somatic hybrid or hybridization. Next is just last one is just suspensor culture. This is also will be the cell culture, or otherwise we call for the suspensor culture. We are using the small or aggregated cells. We can grow in vitro culture in the liquid medium, that is we call for the cell suspensor or cell culture. This is used for secondary metabolic. Large amount of the secondary metabolics we can get through the suspensor culture. What are the strategy will be the suspensor culture is the important strategy. Next we can tabular column given our textbook for the what are these secondary metabolics we are obtained from these plants through this plant tissue culture. One of these decoxing, codeine, capsaicin, vincristine, quinone, that is a medicine given our textbook for the 1 mark question based, 2 mark, 3 mark question based in given our textbook. Next this is a part of the test portion. The type of this tissue culture you can study well, prepared well. Otherwise, I added some slides for your reference. Thank you for watching.